Concept 44.3. Diverse excretory systems are variations on a tubular theme. So there are going to be tubes that are going to be associated with the process of osmoregulation. So here we're really just going to start to think about osmoregulatory organs. So the excretory system is responsible for regulating solute movement, and that can be between internal fluids as well as the external environment. So we'll need to think about what do we want to retain versus what do we want to let go. And when it comes to the process, as is often the case for a lot of the systems that we discuss, being able to utilize the excretory system in the appropriate way is essential for homeostasis, because we're thinking about solute balance, we're thinking about water balance. And while this might seem like we're just thinking about how do we create waste, the role of solutes and water within our bodies has roles that go far beyond the process of just producing urine. When it comes to the excretory system, the goal here is ultimately to kind of modify something that is referred to as a filtrate. So when we first start the process of excretion, we are generating filtrate and modifying that filtrate. And then when we're done with the modification process, then it gets released from the body as urine. And there are three key processes that are involved in the excretory system. So the first is filtration. So we have here glomerular filtration, and we'll talk about that in more depth later on. But filtration would be fluid in the blood being filtered into the tubule system, and it's going to be leaving cells behind. It's going to leave large proteins behind. And so the filtrate is going to be made up of water and blood solutes. And some of those solutes are going to be things that we want to get rid of, but some of them are also going to be things that we want to retain. But we can't discriminate between that at this point in the process. So the filtrate gets modified by the rest of the kind of components, or the kidney ultimately, to produce that urine. Now, the next stage would be tubular reabsorption. And what you should focus on here is the direction of the arrows. So for reabsorption, this would be selective movement of things that are important to the organism or important to us. So solutes like glucose, amino acids, certain ions, we want to move them out of the filtrate and into the extracellular fluid, where they'll eventually go back into the bloodstream, and they'll enter the bloodstream through peritubular capillaries. And this process can be done in an active way or a passive way. The specific mechanisms of this transport, we don't go through within principles of biology. But if you end up taking anatomy and physiology too, that is something that we discuss in more detail. Now, the third major renal process is going to be tubular secretion. And so we see the arrow is flowing in the opposite direction here. So this is going to be movement of substances that are found in the blood into the extracellular fluid and then eventually into the filtrate. And so the idea here would be, when we were creating our initial filtrate during glomerular filtration, there were some things that didn't get in that need to get in. So this is kind of like, let's add in a few more things into the filtrate before it exits out of the body as urine. So this is going to be subsequent modification of the filtrate after glomerular filtration occurs. Here we have a simple filtration mechanism that utilizes tubules for excretion. And this is going to be the protonephridia, and the organism that we're seeing this in is the flatworm. And when it comes to protonephridia, there are a ton of branching tubules that will then filter fluids from the body cavity. And what we find is that these tube-like networks will kind of end with the formation of a flame bulb, and the flame cells will be able to draw fluid through the openings that you see there, and that will be the result of kind of cilia movement. So the cilia movement will allow for the extracellular fluid to be drawn through the openings of the flame bulb. And as the fluid makes its way through, a lot of the solutes will be reabsorbed, so heading back into the body rather than exiting out. And whatever excess water and waste there are will go ahead and exit out of the body through kind of these pores. So there will be openings that allow for that process to happen. And what's seen within the flatworm is that a lot of the nitrogenous waste will diffuse across the body surfaces into the surrounding water. And what we can kind of connect this with is the earlier content. So the protonephridia are going to be osmoregulatory organs, and the urine of flatworms is hypoosmotic relative to the body fluids, which is beneficial due to its freshwater environment. In invertebrates like the earthworm, there are a system of tubules that open both to the inside of the body as well as the outside of the body. And these structures here are metanephridia, and they obtain fluid from the body cavity, so the coelom that's shown here. And they'll funnel that fluid through the kind of tubule network that we see here in order to form filtrate. And things that are meant to stay can be reabsorbed from the collecting tubules, and the fluid that remains within those tubules can be released through external openings out into the environment. Now, when it comes to earthworms, which fall under the phylum of annelids, many annelids are going to live in watery environments, and so they'll release hypoosmotic urine as well as the flatworms that we discussed in the previous slide. For the insect excretory system, there are Malpighian tubules, and these are a bunch of tubes that arise within the midgut, and we can see that they extend through to the hindgut, and they'll be surrounded by the hemolymph. As a reminder, the grasshopper has an open circulatory system plan. And when it comes to the tubules, these will be extensions of the digestive tract, and they'll collect water and waste from the circulatory system, and salt and water and waste will enter the tubules, and certain ions can help to generate an osmotic gradient during this process, and that osmotic gradient draws water into the tubules. So fluid will move from the tubules into the hindgut, and that's where a lot of the solutes and water will be reabsorbed. So you can see here reabsorption of water, ions, and valuable organic molecules. And the nitrogenous waste and excess ions and whatever other waste components there are, they'll get released together with feces through the anus. So this process is a little bit different, in terms of thinking about the way in which excretion occurs. So this is a little bit different from other invertebrates because this is really focusing on secretion rather than filtration of body fluids. So when it comes to our discussion of the urinary system, we'll place the bulk of our emphasis on the kidneys, as the kidneys play an important role in maintaining balance within our bodies. And this can be through regulation of water volume, concentration of solutes that are found within that solvent. And when it comes to kidneys, they are responsible for the formation of filtrate, and filtrate will eventually leave our body in the form of urine. So when it comes to the urinary system overall, we're thinking about filtrate formation, production of urine, transport, temporary storage. And when it comes to the different components of the urinary system, what you have is what is shown. So what you're responsible for is what you have in this image. So we'll spend a lot of time on what the kidney's role is going to be and focusing on the microscopic anatomy of the kidney as well. So we're thinking about how are we capable of forming filtrate within the kidney. When it comes to the ureters, these are paired tubes. And these are going to be transporting urine. Once it leaves the kidney, its form will be referred to as urine rather than filtrate. So it will transport urine from the kidneys to the urinary bladder. And then eventually that urine will exit out through the urethra. We won't put a lot of discussion into specific types of vessels that will be associated with the excretory system in mammals, but I just also want to add in, just because they're here in your illustration, major vessels like the aorta and the posterior vena cava are just shown here, but then also the renal artery and renal vein would be examples of vessels that are going to head towards the kidney and then away from the kidney, respectively. The reason why I added this particular slide is because the example of the mammalian excretory system that's shown within the Campbell book focuses on the female excretory system. So I just wanted to give a sense of the differences that are seen between the male urinary tract and the female urinary tract. And I'm not really going to go through, like morphology and form and function of the different components. I really just want to focus on the urethra, the pathway in which the urine will be expelled, just to show that the length of the urethra in the female is significantly shorter than the length of the urethra in the male. So when it comes to the male, it starts up at the prostatic urethra and ends with the spongy urethra, ultimately exiting out of the urethral orifice. And the reason why I bring this up, if we think about it from form and function, combine it with your knowledge of pathogens, is that when it comes to the female urinary tract, as a result of kind of the short distance that bacteria needs to travel in conjunction with the proximity of the urethral orifice to the anus of the female, it's far easier for proliferation of bacteria to occur. So meaning, things like urinary tract infections occur in much higher instances in females compared to males. And that really just has to do with the kind of length of travel that would take place in one versus the other. And if not treated, then bacteria can continue to proliferate, work their way upwards. You can have, infection of the bladder as a result, or you can continue to work your way upwards beyond the ureters and have kidney infections if things aren't taken care of. So, the point of this image, because this one is from a different Pearson book, is just for you to notice the difference with respect to the length of the urethra and think about how that might play a part in urinary tract infections or in the movement of urine out of the body. Now, when it comes to the kidney, the main role is excretion, but it can also accomplish things like regulation of blood volume, blood pressure, blood osmolarity, and this is accomplished through regulating how much water we release versus how much water we retain. And in addition, the kidneys can release a hormone, erythropoietin, often abbreviated as EPO, and that helps to stimulate the production of erythrocytes, and that helps to facilitate oxygen transport. And this is just a few things that the kidney helps to accomplish. What we have within this slide is some internal gross anatomy, and this isn't as detail-oriented as other illustrations, but we'll just focus on what's being provided for us within the Campbell book. So we have a frontal section, and within this frontal section, we are able to see 3 distinct regions of the kidney. And that is the renal cortex, the renal medulla, and the renal pelvis. Now, in terms of kind of dissection terminology, the most superficial portion is the renal cortex, followed by the renal medulla, then the renal pelvis. Now, essentially renal cortex and renal medulla, they make up the urine-forming portion of the kidney. And that's what we'll talk about the most. So when we think about our functional units of the kidney and the events that are taking place, we're going to be looking at the events that occur within those 2 specific regions. The third component, the renal pelvis, is a funnel-shaped tube, and it is continuous with the ureter. At the point that the fluid enters the pelvis, it's referred to as urine. So urine is going to flow out of the renal pelvis through the ureter and then move into the bladder for temporary storage. And all the movement that's occurring at this point takes place through involuntary muscle contractions, and it's smooth muscle, and the process is called peristalsis. And that's an event that we'll talk about again when we get to other discussions later on in the semester. Now, when it comes to the kidneys, it also plays a role in cleansing the blood and adjusting the composition of the blood. So as a result, it has a rich blood supply. And there are large renal arteries and there are renal veins. And the renal veins, when they exit out of the kidney, they will empty into the inferior vena cava as a way to link this to the circulatory system discussion that we've had earlier. So this is essentially the major components of the kidney. And then we'll start to zoom in and think about these smaller components that we find within this organism as well. So this picture is showing us a zoomed in portion of the kidney. We can see that this area is the renal cortex, this area here, the renal medulla, and this is showing us the nephron. And the kidney has more than 1 million nephrons. And when we think about kidney function, what we're really thinking about is nephron function. So the nephrons are the structural and functional units of the kidney. And what this means is that it's the smallest physical structure that's capable of carrying out the function of the organ. So to understand how the nephron works, as they often say, is to understand how the kidney works. So each nephron is responsible for creating a cell-free and a protein-free filtrate from the blood. And from that filtrate, there can be recovery of chemicals that the body needs and release of chemicals that it does not need. Now, in looking at this image, we can see that there's two different types of nephrons. There's a cortical nephron and the juxtamedullary nephron. Now, the cortical nephron accounts for the majority of nephrons that are found within the kidney. And apart from a really small portion, the majority of the cortical nephron is found within the cortex. It has a short nephron loop, and we'll go through the different divisions of the nephron in a later slide, but the nephron loop is pretty short, and the glomerulus, which is the structure here, is pretty high up, so it's far away from the boundary between the cortex and the medulla, so sometimes called the cortex-medullary interface. The juxtamedullary nephron plays a pivotal role in the kidney's ability to produce concentrated urine, which is a concept we'll talk about in a little bit. And this process helps to conserve water. Now, the juxtamedullary loop, when we compare it to the cortical nephron loop, is significantly longer. So we have a much longer nephron loop here. And when we take a look at the glomerulus, it's a little bit closer to the cortex-medulla interface. So when looking at this illustration, something that isn't currently identified because we are focusing on the nephrons here is the collecting ducts. So down here are your collecting ducts, and if you count them, you'll find that there are 5 of them. And the reason why I draw your attention to it now is that if there are 5 collecting ducts down here, and you take a look at the region in which you find the nephrons, you will find that there's far more than 5 nephrons depicted. And so this gives us a sense of the relationship between the nephron and the collecting duct. So there will be a multitude of nephrons which will generate, process, modify, filtrate, and they will kind of collectively add that filtrate into a collecting duct. So a collecting duct can receive filtrate from multiple nephrons rather than a single nephron connecting to a single collecting duct. And I just say this now because a lot of things are illustrated in a way that may make you think that a nephron includes the collecting duct, which does not, and B, that a single nephron is going to feed into a single collecting duct. So when we say that there are millions of nephrons, we would be saying there are millions of collecting ducts, and that's also incorrect. Here we have a visualization of nephron organization. When it comes to a nephron, they will all have a renal corpuscle, and a renal corpuscle is composed of a glomerulus and a Bowman's capsule. and this is continuous with the renal tubule. And the renal tubule consists of the proximal tubule. It consists of the nephron loop, which is often referred to as the loop of Henle. It is composed of the distal tubule. So all of that is renal tubule. And the collecting duct, which is shown here, we're not including under the category of renal tubule. Okay, so while the collecting duct is shown kind of here in an obvious way, in a conspicuous way, it is not a part of the nephron. Okay, so some books may describe it as such. We are not doing that. So your nephron is glomerulus, Bowman's capsule, proximal tubule, loop of Henle, distal tubule. Okay. And as a reminder, there will be multiple nephrons that will divert their filtrate into a single collecting duct. So it is not a one-to-one relationship. Now, in looking at this particular illustration, and I'm hoping that it is not the case in the student textbook version, that's just the instructor slide deck, but the arrow for the arterials are not entirely accurate. The efferent one is, so there's an efferent arterial, which is going to be exiting out of the glomerulus, and then there's an afferent arterial, and the afferent arterial is red here, but the line that it's showing looks like the kind of tubule portion. Okay, so the afferent arterial arrow is not correct here, so it should be pointing to the red vessel. Okay, so there's an afferent arterial from the renal artery, and there is an efferent arterial from the renal artery. When it comes to the efferent arterial, what they supply will be different depending on the type or category of nephron you have. So if it is a cortical nephron, the efferent arterial supplies the peritubular capillaries. If it is a juxtamedullary nephron, the efferent arterial supplies the vasa recta. Okay, so that's something that is just another way to distinguish between the two different categories of nephrons. So visually, your Campbell textbook doesn't go into enough differentiation between peritubular capillaries and the vasa recta. But for peritubular capillaries, they are going to arise from efferent arterioles, and efferent arterioles have a pretty high level of resistance. So as a result of that resistance, there's going to be kind of diminished pressure because the blood has to deal with that high level of resistance. So by the time you get to peritubular capillaries, the movement or what you'll experience in that location will be relatively low pressure. So as a result, these low pressure capillaries are capable of readily absorbing solutes and water from the tubule cells as the substances are reclaimed from the filtrate. And when it comes to the renal tubules, they're really closely packed together. So the peritubular capillaries of each nephron are able to absorb substances from several nearby or adjacent nephrons. Now for the vasa recta, you can see here where you find it, which is in really close proximity to the loop of Henle. These are going to be long, straight vessels. In contrast, these kind of like meandering vessels of the peritubular portion. And they run parallel to the nephron loop. And you're going to find them in the juxtamedullary nephrons for reasons that will become more clear as we progress forward. And when it comes to the vasa recta, they supply oxygen and nutrients to the tissues that they pass through, but they also play a really important role in the process of forming concentrated urine. Okay, so your book didn't provide an illustration for that. I'm going to give you one from a different book. But when we think about urine formation, whether it's concentrated or dilute, it is really important to incorporate the role that the vasa recta is playing in helping to accomplish this.